Good morning students, welcome to online biology class and the today's topic is continuity about the fundamental unit of life and today in this session we are going to be discussed about the nucleus. Yes, in the last class we already discussed about the cell wall and we already discussed some prokaryotic and eukaryotic and we did that experiment. Which experiment we did? The onion cell and the rheolith. In the onion cell, we discussed about that onion cells. The cell is going to be composed to form an organism. To derive that, we did that activity, the help of onion and a microscope. Yes, and the rheolith, we discussed about that the osmosis. The function osmosis is going to be occur. The absorber of water will be happening in the living cells compared to dead cells. We discussed in that experiment and also we discussed about some solutions. Hydrotonic solution, hypertonic solution, hypotonic solution, isotonic solution. These and all we discussed in the last class and also we discussed about the plasma membrane also in the last session. And today's class, in the last class when we are going to be observe the cell wall of onion, we observe the structure of honey. We observe the honeycomb-like structure. Yes or not? This honeycomb structure we observed with the help of one dye. That the dye is nothing but iodine solution. With the help of this iodine solution only, we can see that the cell. Why? The cells are not having any color. They are transparent. The cells cannot having any color. That's why for our identification we are going to be add the dyes to see the cells. Yes. Now I draw the thick line will be observed, light thick blue color when you observe under microscope. And here the dotted structure I showed to you know. That structure is nothing but the cytoplasm. This cytoplasm we are going to be discussed in the next session also. Yes. And I draw some small round-like structure. These are nothing but the nucleus. Yes, that the nucleus only we are going to be discussed in this session. Who discovered the nucleus? Nucleus was discovered by Robert Brown. After the Robert who discovered the cell, Robert Brown discovered about the nucleus. In the cell, something was there. That's why it is going to be starts divided. Like that, started his experiment. Not only he stopped with experiment with a few things. He extended that experiment on plant cells, living cells, dead organisms, dead plants, and he concluded that the nucleus plays an important role and it is very crucial for a cell to perform the cell division. He only concluded that. Yes, and in this session we will discuss what the nucleus may have in content, where the nucleus will be present, what does the function of nucleus we will see in this session. Yes. The nucleus, how the structure will be nucleus? The nucleus will be present in a center part of a cell and it is going to be formed with the help of double layered membrane. This is double layered membrane. Nothing but we may call it as a nuclear membrane. Yes, this is nuclear membrane. It is filled with some amount of fluid. Yes, nuclear fluid, that is fluid. The middle of the nucleus, we may call it as a nucleolus. Nucleolus. It may contain the nucleons. It's contain the nucleides. Yes. These round compact like structure, we may call it as a inside the nucleus nucleoid. It may contain the chromatin material. What does it contain? Chromatin material. These chromatin materials are having chromosomes. What it is having? Chromosomes. These chromosomes structure will be rod-like structures. Chromosome structures are rod-like structure. Rod-shaped structures. Yes. And next, the nuclear membrane plays an important role in the cytoplasm for the entry of any material to inside the chromatin and exit materials to outside. This nuclear membrane is going to be act as transferring. Yes, these chromatin materials is having contained the DNA and proteins. What does it may contain? DNA and proteins. DNA means nothing but deoxyribonucleic acid. Deoxyribonucleic acid. Yes. When the chromatin material wants to divide, the chromosomes will get a position rod shaped. In a cell itself, it will be divided a rod shaped. How the chromosome structure? The chromosome structure will be like this. Yes. This is the chromosome structure. Here, the telomeres will be present. Telomeres. We will discuss about the telomeres and about the cell division. We will be in the next session. The cell wants to get divided. It will be divided like this. It will be one cell and this will be one cell. Yes, like this the cell will be divided. The chromosomes divided. These DNA is nothing but the DNA molecules. What does actually the chromosomes contain? The chromosomes are having the DNA molecules. The DNA molecules are the constructing and it's necessary for carrying the genes. For carrying the genes. Yes. What does mean by genes? Genes are nothing but inheritance. Inheritance of characters of parents. Characters of parents carrying to next generation. Carrying to next generation. That's why you may call it as a genes are only the hereditary characters to pass the characters from one generation to another generation. How we are getting the characters of from our father's mother? How? Like that the genes are present in the DNA nucleus. That's why the characters same like a father, like a mother, we are going to be getting by the help of nucleus. That's why the nucleus is only the crucial role. It is very important for us to share the characters from one generation to another generation. These and all DNA and proteins all will be present in a chromatin material. Yes. And next we will be discussed we already told one cell is going to be divided into two cells with the help of mitosis and meiosis cell division. That we will see in the next class. Here, the cytoplasm only going to be playing a role to divide these cells. And next, the prokaryotes. The prokaryotes are not having nuclear membrane. Nuclear membrane is not there in the prokaryotes. That's why the nucleides floated in cytoplasm. Floated in cytoplasm. At that time, that's why the cytoplasm of prokaryotes are not having any cell organelles. Only the nucleus only occupy the space. And that's why we may call it as a single nucleus only will be present. Single nucleus only will be present. And when you go to the eukaryotes, nuclear membrane will be present. That's why the cell organelles also will be present in the eukaryotes. These prokaryotes are nothing but bacteria. Bacteria only, no? Best example for prokaryotes is bacteria only. In these, when it goes to the plant, if any plants it may having prokaryotic character, normally the prokaryotes are not having any cell organelles, but the chlorophyll will be present. The chlorophyll will be present. Why? It is a plant. These chlorophyll are not having plastids. That's why the prokaryotes plants cannot perform the photosynthesis. Why? Because the chlorophyll is there, but it is not having the plastids. Plastids are only having the capable to trap the solar energy. That's why the plants of prokaryotic cannot perform the photosynthesis. How does the eukaryotic plants can perform photosynthesis? Yes, and these nucleus we discussed in this session, and I told you with the example of one activity. That activity is nothing but about the human organism is having the nucleus. Students, we will be continue with the activity. We are going to be aim. Observing the nucleus cells in human cheek cells. We are going to be collect the saliva of the humans. Yes, from your mouth we are going to be collected the saliva. Yes. What are the materials needed? Saliva, iodine solution, spoon, slide, cover slip, microscope, burner, etc. Alcohol also needed. Yes. are going to be perform these with these activity procedure. First of all, the person who is going to be who are going to be collected the saliva, that person needs to be close the mouth up to 10 minutes, not to take release the saliva outside, not to inhale the saliva to inside of their body. That person needs to be store the saliva for 2 minutes. After that, they needs to be collected with the help of the spoon. They will keep the spoon, open the mouth of that person. Spoon will be touches the cheek. Inside the cheek, they will be scrubbed. And the spoon may contain some amount of saliva. Yes, this saliva we need to be placed in a slide. This is the slide. We need to be placed on a slide. Need not to dry. That slide may contain a thin layer of saliva only. Don't be having any bubbles on them. Okay, na? A thin layer of saliva needs to be collected on a slide. After that, this slide we are going to be pour this slide need to be burning. Just we need to be placed it near the fire. Just kept it and remove it. Why? Because the cells needs to be attached to the slide. After that, remove the slide outside. Pour the iodine solution. 2 drops only. 2 drops of iodine solution should be poured on the saliva. After that, 2 minutes. Wash with water. Wash with water. After washing the slide, you need to be add the alcohol. Alcohol also 5 drops. 5 drops of alcohol should be added. After adding alcohol, let it be dry. Drying after, rinse with water. Kept the slide under water. After that, you need to be add the cover slip. Cover slip is nothing but a small glass slide only. That glass slide, the cover slips will be in a round color, square shape. That cover slip without the air bubbles, we need to be add it on the cell, cheek cells, with the help of a glue. The glue will be there. You need to be take the precautions. What precautions you need to take? When you are going to be close with the help of a cover slip, you need to be pressed at one side that cover slip. Slowly you should be placed it on the other side, not to enter any air and not having any air bubbles inside the cell when you are placing the cover slip. That should be, must be careful when we are going to be adding the cover slip. After that, you will be adjusted to the microscope and observe that how the structure will be like a honeycomb only. But the round color, the nucleus structure will be appear in a blue. Why blue? We added iodine solution, no? It will be look like in a blue color. That is about the experiment we are going to be performed to observe the nucleus cells in a human cheek cells. That's why in every part of our body is having the nucleus. The cells are having the nucleus to divide. Not only a cheek cells. When you pluck your hair to that, the tip it is having a white color round bubble. That bubble are also having the nucleus. The full organism is going to be divided with the help of the nucleus only. Yes children, today's session is going to be completed, and in the tomorrow's session we will discuss about the differences between prokaryotic and eukaryotic cell. Thank you children.